Yesterday we have seen that one of the things people are doing is making populations fragmented, and that many organisms now have to live for that reason in rather small populations that may be badly connected to other small populations. And that may result in a number of genetical consequences. So one of them, which is, or no, you can summarize them by saying there is a loss of genetic variation. And the way it works is that animals start inbreeding because they will be often in groups where there are a lot of relatives because they're in a small population. And so they mate with their relatives, and this leads to inbreeding. And yesterday we have talked about metapopulation dynamics, and we've seen also that empty patches, they can be recolonized by a small number of animals. And when they do so, there are so-called founder effects. And finally, because of the fact that animals have to live in small populations, there are random processes, and those are known as genetic drifts. Founder effect is also a stochastic effect. And the consequences of all those things is the loss of genetic diversity. And now you can ask yourself, is that so bad then? And yes, that is bad because it leads to direct fitness loss and to a long-term loss of evolvability. And that's what I want to talk about today. So we'll talk about inbreeding, founder effect, genetic drift, and about how this results in fitness loss and what the consequences are for evolution. So this summarizes against, but it defines inbreeding. Inbreeding is the reproduction from mating of pairs who are closely related. Now the consequences of inbreeding are loss of heterozygosity, generally because we have two copies of each chromosome. There can be different alleles of the same gene on these two different chromosomes. And so if there's something wrong with one of those alleles, then you have an insurance because there's still the other allele. Now, same way of saying loss of heterozygosity is increase in homozygosity. So that says more and more loci will have the same allele. And so then there is no rescue effect anymore. And very often, because we have different alleles, we can have deleterious alleles, often, say, because of a bad mutation, and then a gene doesn't work properly anymore. And so once you become homozygous, then you get those bad genes expressed. They could even be lethal, and then you die. Or it makes you ill, and then you have a low fitness. So there are fitness costs of inbreeding. So here you see the effect of inbreeding. So you start with two chromosomes that come from the male and the female. And in this case, all the alleles are different. So you have 100% heterozygosity. And then after 5 generations of inbreeding, then you arrive at 100% of homozygosity. So in small populations, this could go quite fast. Now the cost of inbreeding, in terms of fitness, they are very difficult to measure in the field. And so people first, they have looked at zoo populations of animals, and they found that these populations, they're also often small populations, because a zoo is also an island, and although zoos can exchange animals, there could be inbreeding. And what you can do then is you can assess the fitness loss in terms of how many of the young die because they don't have fitness. And when you do that, you see indeed that there is a cost of inbreeding, and it even looks quite high. But at the same time, you have to think about the fact that an animal in the zoo, it gets its food, it has no enemies. If when it's ill, there is a doctor coming. So it lives under what we call relaxed selection. There's no natural selection working on the animal anymore. So even under that situation, having a fitness cost means that in nature the fitness cost should be much higher. And indeed, Derek Roth and one of his postdocs, they have shown that they've looked all the literature and tried to find estimates for the cost of inbreeding in the wild, and they arrive at a value which is more than 6 times higher than the cost of inbreeding in these zoo populations. And such a high value means that it's really biologically very important. So once you get small populations, then it's difficult to save them because their genetic quality decreases simply by the fact that they live in these small populations. And there are people who have shown that this is a consequence of homozygosity, and they can show, for instance, in a butterfly population, they measure that lives on small islands, so they're all small populations. It's a really meta-population effect, and so they can catch these butterflies and then measure the heterozygosity, and they can also, because they follow these butterflies over years, they know which populations go extinct. And what you see is that the populations that have a low heterozygosity and hence a high homozygosity, those are the ones that have a much higher rate of going extinct. And one of the things that happens is this expression of bad genes. And so you have these animals with a lower fitness that tend not to reproduce or to die early. And here you see an example. It leads to developmental instability. And here you see the hands of a person that has one normal hand, and the other hand has 6 digits. And you see that it looks not very good. It doesn't look really functional, the left hand. Now, yesterday I've lectured you on ecological effects of living in small populations, and then the conclusion was also that small populations can go extinct by stochastic processes because of variation, stochastic variation in the environment. And so the question is now we have 2 explanations, and so we can ask ourselves, is the genetic deterioration of population, is that more important than the stochastic variation of the environment, or do they both play a role? And so here are people, Wooten and Pfister, and they have published an article this year in which they in fact show that these small populations of seaweeds, here you see a photograph of these seaweeds at low tide, so at high tide they're underwater, and they looked at the demographic effect of these weeds being in small populations. So they made populations of different sizes, and then they can show that the ones that have a low genetic variation, they go extinct much sooner. But then when they compare the rate of extinction or it is the time until extinction of, and that's the lower graph, inbred left and outbred populations right, and the control population in the middle. And the black columns, those are the effects of the genetics, and the white columns are the effect of stochastic variation in the environment. And so you see that, oh no, it's the other way around. The genetic deterioration is less important than the stochastic effects of the environment in this case. But in other cases it could be different. And both play a role, and you could still say, well, even if the environment is very important in making animal populations going extinct, then those that still survive, they will have even a harder time maintaining their genetic variation, and they will go extinct in the long term anyway. So this research does not mean that you can ignore the genetic deterioration. Now, I've mentioned already two words, genetic drift and founder effects. And the idea of genetic drift has been coined by two very important theoretical biologists in the last century, Sewell Wright and Ronald Fisher. And you know Ronald Fisher not only as an evolutionary biologist, but also as one of the fathers of statistics. So he was a very clever man. But Sewell Wright probably was as clever as that. He still published scientific articles when he was 99 years old. So what is genetic drift? It's a change in the genetic composition of a population due to change or random events. And so what happens is that there's always mortality in a population, and that not all the animals reproduce. And the effect of that is that not all the variation of alleles is taken to the next generation. And that happens more in small populations than in large populations. In very large populations, there are so many people, so many animals contributing to the next generation that it's highly likely that all of the alleles will be transported to the next generation. So I should warn you that a loss of an allele is not due to selection. That's another possibility, and bad allele can be taken away by natural selection. This is just a random process. Bad luck for the allele. Now, and the founder effect is very much the same, in fact. Ernst Mayr invented it, but you should remember, I think that's the important message, because you will see these words in the literature quite often, is that when you talk about loss of alleles due to random processes in time, you talk about genetic drift, and when you talk about space, then you talk about founder effects. So let's look a little bit more. So here you have Sewell Wright and Ronald Fisher. And here is a simulation experiment to show the idea. So you have bottles with marbles, and then you can choose to have blindly some of the marbles, and you make pairs of them, and then you let them get offspring, and those you put in the next bottle, etc., etc. And when you do that for a number of generations, again you arrive at a homozygous population. In this case it's all blue, but if you would do the experiment yourself, you might as well end up with all reds, or you might end up with blues that have 3 or 4 dots on it. That's all possible, because it's a random process and the answer is always different. And here you see the founder effect. In fact, it's the same. So, but now we talk, because Ernst Mayr talked about that in relation to animals on islands and why very often new species come into existence on islands. And what he says is that so the left, where we have 20 blue and 10 red marbles in this case, is the mainland, and at the right you see the island, and there are 4 immigrants, and they are chosen by chance. And then what you see is that all of a sudden there's still both red and blue animals in the population, but the frequency has changed enormously without any selection. And that's what you call the founder effect. So on the island, unless there is strong positive selection for the blue one, the red allele will stay in the majority, and that's very different from the mainland, and therefore these populations can then develop in a different direction. And here you have Ernst Mayr, also one of the very influential biologists of last century. Here is a graph that is the result of a computer model, because working with bottles and marbles, that's rather tedious and it takes a lot of space and a lot of time to do so. So it's better to calculate what happens. And so what we see here is the genetic diversity on the y-axis, and then you can see what happens with different population sizes. Now you see small populations, they go down in variation very rapidly, and then the larger, so you have a population of 10 that really goes down to 0 quite rapidly. 50 takes more time, and there's still some variation left at the end of the calculations. That is after 200 generations. But note that even if you have a population of 200 individuals, that the genetic variation is going down. And the reason I want you to note this is that we all know that there are less than 200 tigers in Nepal, and that these tigers, they live in isolated populations, and so it is inevitable, if we don't do something, that the genetic variation of the tiger population will go down. So we have to do something. Here is a graph that's sort of also showing genetic drift at work, but now you can follow what happens, and you can see, in fact, that in small populations, that's the upper graph, that the stochasticity, it's much higher. You see very rapidly in one population you see an allele coming to fixation, so that goes very rapidly to 100%. In other populations you see alleles disappearing very quickly, going down to 0. And the others you see that they fluctuate wildly. And then when you increase population size from 10, 20 to 100 to 200, you see that the stochasticity is decreasing. And finally, at a population of 2000, you see that there is a good chance that all the alleles stay in the population. So that tells you something about the population size that you need to maintain the genetic variation. So to say it in other words, we would have to aim for 2,000 tigers. And here is another way to show it, and I like to do that because in the past we've talked about connectivity between patches and that you could increase it by corridors. And so in these graphs you can see the effect of connectivity. You can see that what this graph shows on the y-axis is how populations have diverged from each other. And that's because they're small. And you see that when the connectivity is low, there's more isolation. And you can also see that when the islands in which they live, when the area is small, that there is also more divergence between the populations. So they're drifting away from each other. And when there is a lot of connectivity, and when the surface of the patches is large, you see that they do not differ much, which means that they all share a large amount of genetic variation. Now, so far we have only talked about stochasticity and not about selection. And so I want now to introduce also an evolutionary view on this problem, which is, and I can do that by telling you the story of the Red Queen. An English author, Lewis Carroll, he wrote first a book called Alice in Wonderland, and that was quite successful, and then he wrote a sequel, and the sequel, the title is Through the Looking Glass. And in that book, Alice arrives in a country that is governed by the Red Queen. And the Red Queen is in fact a piece of it on a chessboard. It is a chessboard queen. And she's quite surprised to find that this queen is running like mad. And she comes from a world where when you stand in a place, you stay there, and sometimes you want to stay where you are. And in fact, that's what we also want with our animals. We have tigers in forest, and we want that the tigers stay there forever. And then the queen says, to explain to Alice why she is running, she said, "Here it takes all the running you can do to keep in the same place." And now we have to ask if that is true for nature also. And the person that did that was Lee Van Valen. He's an American scientist who sent ground proposals to do field ecological studies that were all refused by NSF, and so he had to decide to become a theoretician. And now he's very famous because he studied the literature and he looked at extinction rates over geological times. And so the idea is born not in ecology, but it comes from sort of extinction rates of taxa over geological times. And he noticed that the extinction rates over time are constant. And that's what you see in the graph at the right side of the photograph of Lee. And then he started to think why this could be. And his hypothesis was, and that's important for us, that in the long term, every organism runs out of genetic variation enough to evolve. So and then he formulated this Red Queen hypothesis that said, in fact, to survive as a species, you have to adapt yourself the whole time, and only then you can stay in place. And the reason for that is maybe not because of climatic changes or things like that, but the most important aspect of the environment, those are other organisms. We, as we sit here, we are in a constant battle against bacteria. Our immune system, we have to invest in the immune system, has to battle all these bacteria that try to attack us. And sometimes a new illness comes in a population, and it takes time to build up new resistance, and many will die, but maybe the species can survive because some of the animals will become resistant. So you have to continue to adapt yourself. But in the population, these challenges always cause mortality, and with mortality, some of the alleles will drop out of the population, and then over geological time, you go extinct anyway. So we, as we sit here, we will have hopefully a very happy life, but maybe not our grandchildren, but many generations after that, humans will go extinct as well. That's the sad message for this morning. So also organisms have to run. They have to evolve constantly and to adapt to survive in an ever-changing environment. And in particular, against other evolving organisms, that makes that this challenge cannot be solved for once and forever. They're challenged the whole time, and pathogens parasites and predators play a role there. And now we go back to the small populations, but because we know already that small populations tend to have low genetic variation, and so small populations, they fall more easily victim to the red queen, as they can no longer evolve and adapt to the changing environment due to the loss of genetic variation. And then they will lose even more genetic variation because of the red queen. So it's really a pessimistic story I tell this morning, but I thought maybe for the first talk in the morning, when you're fresh, you can deal with it psychologically. Now, can you show that the red queen hypothesis works? Yes, you can. Here is an example where you can show that inbred lines of plants suffer more from herbivores than outcross plants. So that's an example of the red queen, because it means that the inbred plants then are under selection of the herbivores, and those genes will be, those individuals will be washed out of the population, and then it goes on and it goes on. Another way to do it is look at and compare animals that reproduce sexually, because sexually reproducing organisms, they have recombination in the genes, so they shuffle the genes of both parents the whole time, and therefore they can create new variants which are a challenge for the bacteria and the other pathogens. And in this case, people have looked at parasites that live in fish, and these fish, they live in the same sort of sources, it's small ponds in America. And one form is sexual, you have males and females, and there is another form which has been derived from that species, and by some mystery they have succeeded in the females getting female offspring without mating with males. So they have become clonal. People have not yet invented it, that's why there's still males in the audience. And now you can compare with these fish the parasite loads, and those are on the y-axis of sexual and asexual populations. And you see that the sexuals, those are the lower curves, they do not suffer so much of these parasites, and so that's a good example of the red queen hypothesis as well. And so, to summarize this rather somber picture I'm giving you, once populations get small, then they become prey of stochasticity, inbreeding, genetic drift, and so they lose genetic variability also because of the red queen, and so they spiral down until they disappear in extinction. Can you do something about it? Well, here you see an example of a bird that lives on the North American prairies, a prairie chicken, and there was one population that was small, and so it became inbred, and then after a while you see that the fertility goes down, so they lay eggs, but many of these eggs do not hatch, so they get fewer and fewer children, and they become more and more homozygous, and people realized that they were losing this population, and so then they decided to bring in animals from another population to increase the genetic variation, and you see an immediate response. The numbers go up because the reproductive potential of the animals is restored. All the eggs hatch, they produce larger number of chicks, And so the recruitment is back and the population starts growing again. So this is also a proof of everything I told you. So the way to extinction is, because all the factors we've discussed yesterday and today, they work together. So you have stochastic variation in small populations due to stochastic variation in the environment. Then there is the LE effect we discussed yesterday, which lowers the fitness in small populations. Then there's inbreeding, which results in genetic drift. And then finally there is the red queen that finishes everything off over a longer evolutionary time. A sad story indeed. But there's things we can do about, like in the story of the prairie chicken. We can try to counteract these things. We can counteract stochasticity by connecting small populations. And so the building of corridors is very important. And we should also save smaller parcels of forest, as I told you yesterday, because empty patches also make that you cannot say there are no tigers in there, let's cut it away, because that also helps the populations to spiral down. And in the worst case, if you cannot improve connectivity, then maybe you should start, like in the prairie chicken case, to load animals from a different population into an airplane and bring them over to the population where the genetic variation has become low. And in fact, if you now look at zoos in North America and Western Europe, they don't want to pay the cost of inbreeding anymore, and so they have breeding programs of endangered animals in which they take care that there is no inbreeding, because they keep lists of all the individuals, they know their genetic background, and then the zoos exchange animals that are flown over from this side of the world to the other side of the world to make sure that the gene pool stays as rich as it was when these animals were collected from nature.
I just have one question regarding the inbreeding. How long is it before you see the deleterious effects of the inbreeding coming into the nature?
I think I gave the answer in the lecture. There was 2 examples where you needed only 5 generations to reach totally homozygous populations. Of course, it depends on the size of the population. So the larger the population, the longer it takes. And if you have really a large population, say an effective population size of 10,000 or so, then it will take forever, and that's what we want. But populations of several hundred animals, the period of 10 years, you can lose a lot of genetic variation.
Is the mutation, and why would the mutation counter the effects of the inbreeding? Does that take too long before inbreeding has the effect, before mutation would counter the deleterious effect of the inbreeding?
Yes, mutation rates are generally quite low, and the majority of mutations don't have any adaptive value. There are very few that have, which help evolution, but there are a lot of mutations that simply demolish a gene, the expression of a gene, and then you have all these things like movable elements in the genome that when they sit in the middle of a gene, it cannot function well. So there are lots of changes in the genome which in fact are very harmful for the organism.
That's true, the inbreeds are more susceptible to the pathogens, that's true. And we have seen in the cultivated plants in the arable lands. But is there any kind of experimental evidence on that? The inbreeds, they have a less resistance, or this is only the theoretical hypothesis?
It's of course much more complicated than I could tell in a very short time. There is evidence, because I showed you the evidence, which is a Datura species, and so there is experimental evidence for it. But then on the other hand, you know that, for instance, if you do, and plant breeders do that, For instance, they want to have cucumbers that are resistant to spider mites. And then they start to select plants that are resistant to spider mites. And once they've finished the selection, the genetic variation in their material has decreased, but then the plants do better against spider mites. But maybe the consequence, and that's my point, is that by making plants resistant to spider mites, they have become more vulnerable for fungi or for aphids or God knows what.